Thank you very much. Sam mentioned my work focuses mostly on getting more stress-tolerant crops for developing countries, and I gave a seminar earlier today talking about that work. Sam said there was this other additional opportunity to talk about climate change a little bit. Of course, getting more drought-tolerant, heat-tolerant plants is part of climate change. But I'm going to take a little bit broader view and talk about some things we don't know about climate change. And this is, I think, one of the main ones that's going to be what I'm calling a wild card, meaning responses of plants can go in unexpected directions because of mineral stress. And I'll define that in a second. Just a second here. So here's what I'm going to try to say today. Most plants on Earth suffer at least one serious mineral stress, meaning not enough of a nutrient or too much of something they don't want. And little is known about these interactions with climate change, although it's likely that there's going to be important interactions between mineral stress and climate change. And I'm going to give you three examples. One is a recent study we completed on soil taxa by elevated CO2. One is phenology and phosphorus stress, and one is the outstanding PhD research of Dr. St. Clair, which I think illustrates some important points. And finally, I'll talk about a conceptual gap that we have in this field. So, okay, for the first point, most plants on Earth suffer at least one serious mineral stress. So here's, and if you see this here, the Earth from space, you see the dark green areas is where most of the plants are. You have the boreal forests and the tropics. And let's take a look at some of the soil conditions in these environments. This is the nitrogen balance. I showed this earlier today in the agricultural seminar. So the brown and yellow colors are negative nitrogen balance, meaning there's less nitrogen each year than there was before. So these are in nitrogen deficit. And there's some areas here, this map focuses on agricultural systems. It doesn't include the Congo and the Amazon. Of course, it's widely acknowledged that nitrogen deficiency is a pervasive limitation to plant growth on Earth. Phosphorus. Perhaps less widely recognized, but also a pervasive primary limitation to life on Earth. Any red color in this map represents not enough phosphorus for plant growth. Acidity, low pH of the soils, usually weathered soils, which have low phosphorus, low calcium, low magnesium, low potassium, typically excess aluminum, excess manganese. It's a huge constraint. Remember where most of the biomass on the Earth was? Those are in acid soils. Why? Because plants need water. And where you have water, you have soil weathering, and you end up with acid soils. So a huge portion of the biomass of the Earth is actually growing in soils with these multiple problems that are driven by acidity. High pH. Now, here the kind of purple color here is high pH, so these semi-arid regions of the Earth, including Utah, have high pH problems, which also create nutritional problems for plants. In this case, once again, low phosphorus, low iron, zinc, manganese, and then salinity or sodicity. So when we combine the pictures I just showed you of the Earth, most spots on the Earth have more than one problem. In fact, here's a little quantification of that in a Venn diagram. This is the area of the Earth of plants that are experiencing low phosphorus, and here's plants that are experiencing acid soils, and you can see in this case, 70% of the acid soils are actually low phosphorus also. Those plants have multiple stresses to deal with. This is an example. This is a study I did some years ago with one particular plant, common bean, and we mapped where beans were in Latin America and what the soils were like in those environments. And this pie represents 82% of the total bean area in Latin America at that time. And so this is low nitrogen, and this is acid soils, magnesium, calcium, aluminum. Look at all the constraints in this slice. Look at here. This is just phosphorus, but most of these slices actually have multiple constraints. I think this is typical. Plants usually are dealing with multiple mineral stresses. Okay, point number two. Relatively little is known about how climate change variables interact with mineral stress. Now, this is a Web of Science search I updated this week. So if we look at the past 20 years, there have been 6,455 papers about elevated CO2 in plants. Of those papers that mention nitrogen somewhere, anywhere, there's a good fraction, 2,600. Phosphorus, there's considerably less, and then all these others, which are quite important, there's actually much, much less. Take, for example, aluminum. As we just discussed, acid soils host most of the biomass on Earth. Aluminum is a major constraint in those regions. Out of 6,400 papers, 17 mentioned aluminum. That's not to say they focused on aluminum. Any place in the article that mentions aluminum would come up as a hit here. It could have been just a nutrient analysis of the soil or something. So my point here is that these are really important, but they're not getting a lot of scientific attention. If people think about nutrients, they think about nitrogen. But there's reason to believe that these things interact with climate change variables. For example, in a high CO2 or changing humidity environment, changing precipitation, temperature environment, that will change plant transpiration, which will change the transport of nutrients to the roots, which will change the acquisition of all of these things. And likewise, I can go down through here and say we're talking about mycorrhizas or exudates. A lot of times there's question marks. We really don't know. But there's clearly climate change variables that are likely to have important interactions with these processes. I'm going to be talking in a minute about the case of metal photochemistry, which is one of the examples I'll be giving. So that was my second point. My third point is that there's likely to be important interactions. And here's where I'm going to be giving you examples. The first example is a kind of a case study. Plant response to elevated CO2 is controlled by soil taxa. This was just recently. It's in press. It's recent work. So this is a soil map of the Earth. As you know, there's a taxonomy of soils, and the U.S. taxonomy system is classified into 12 soil orders at the largest aggregation. And so here's, you can see different parts of the Earth have these different soil types. They're actually quite different chemically and physically and as media for plant growth. This is a picture of what they look like. Nobody had ever done a study comparing how plants grow in all the different soils of the Earth that we know of, and especially as regards to climate change, so we thought that would be an interesting study. So we prevailed upon colleagues all over the Earth to send us soil samples, and yet it wasn't easy with all the permits and everything, but so we got representative soils of all of these soil orders and grew plants in sort of a common garden experiment in one environment. We wanted to grow Arabidopsis in all these soils, but it only grows in potting media. This one is potting media. It grew over here too, but basically it didn't grow in anything, so we couldn't really use Arabidopsis. But we found a plant here, tall fescue, which is a widely distributed grass species, small statured, it's a wild plant, it grows well in everything. So this is the one we use, is tall fescue. And so here's the continuously stirred tank reactors where we can have high or low CO2. This is the leader of this project, Eric Nord, showing how he would go into the chambers. And, of course, you don't want to exhale carbon dioxide, so he had to do this kind of, scuba gear type of setup. And so this was Eric's project. And, of course, we got very large differences in plant growth in these different soils. And the idea was, well, people have many, many studies. I showed you there's over 6,000 studies in the past 20 years have looked at how plants respond to elevated CO2. And in many cases, they say, yeah, plants will benefit from that. That's going to be the silver lining of climate change, is the plants will be fertilized with CO2 and they will grow better, we'll have more food and so on. But those studies were done typically in either artificial media or in some US soil that's pretty fertile, so we wanted to look at all these soils. So here's what happens if we're looking at biomass growth, and here's leaf nitrogen in all these different soils, and the red bars is twice ambient CO2, and the blue bars is ambient CO2. And you see that there was a significant CO2 effect, but it was much smaller than the soil effect and much smaller than the soil by CO2 interaction. So there were a couple of soils. This is one of them, this alpha sol. There was a significant elevation of plant growth by CO2. There's a slight one here. In most cases, there was no effect. They were non-responsive. This is potting media. So these plants loved CO2, and they're growing much bigger than most other plants. Leaf nitrogen, and remember we were talking about how the literature focuses on nitrogen as the nutrient. If a plant ecologist knows of one nutrient, they know about nitrogen. And yet, if we look at leaf nitrogen, there's really no clear pattern of how elevated CO2 affects leaf nitrogen, and it doesn't correlate with how the plants grew. This is the gas exchange data. So now we're looking at whole shoot carbon dioxide exchange rate and also below ground carbon dioxide exchange rate. Once again, potting soil, very big plants, big photosynthesis. Alpha soils, which are fertile soils like we find in Pennsylvania. Mollisols, which are very fertile soils like we find in Iowa. And vertisols, which are very fertile soils, all showed a significant benefit in gas exchange from elevated CO2. The 3 most fertile soils showed a response to CO2. The other soil orders showed no response to CO2. In fact, if we look at how much of the land area of the Earth is occupied by responsive soils, that's in the blue bars here, and the red bars is non-responsive soils. Only 17% of terrestrial land area is occupied by soils that, in this experiment anyway, showed a response to elevated CO2. So what are the points from this study? This was the first study of its kind. In the literature that we were able to locate, the only study that had looked at more than one soil looked at 3. So we looked at 12 soil orders, a wide survey that's been published, and we found the soil order determines plant response to elevated CO2, that the artificial medium gave artifactual results. Only the most fertile soil orders showed a response to CO2, which kind of makes sense. And 83% of the Earth's land surface is non-responsive soils. The principal components analysis, where we took lots of data about the nutrient content of these plants and their endophyte composition and all these kinds of things. So we did a big principal components to see what factors were driving these results. And nitrogen, phosphorus, potassium had really nothing to do with these principal components. The first 4 principal components only explain 63% of the variation, and none of those had anything to do with nitrogen, phosphorus, potassium in any major way. So the kind of simplistic notion we have of fertile soils having more nitrogen and therefore being more responsive is not true. There's a lot of factors in soils that affect plant growth, and these were important in this case. In fact, one of the factors that emerged from the principal components is manganese, which I'll talk about in a minute, which most plant scientists don't really think about. These unresponsive soil orders are not represented in the free air CO2 enrichment face sites, a huge investment by the scientific community in understanding how ecosystems respond to elevated CO2. None of them are on these unresponsive soils. The North Carolina site was originally on an unresponsive soil, but it had been fertilized and limed prior to the study, so it really does not represent. For example, the Amazon, the Congo are not represented by these facilities. Global models that predict, you've seen global models, in fact I'll show you one in a minute, that are predicting how the climate's going to change. They have certain assumptions about how vegetation responds. None of those models consider these kind of soil constraints I'm talking about. They're based upon literature that's focused on those rich country soils, fertile soils, that don't consider these unresponsive soils. So where's the silver lining? Are we really going to get more plant growth in a high CO2 world? Question. Example 2. Phenology regulates plant response to low phosphorus. This is work led by Eric Nord, published in several papers, including some that we haven't published yet. So this is Raul Jaramillo's work. He collected, depending upon the soil types, between 50 and 150,000 sets of profile data. He deserves some kind of a medal for, all of my graduate students deserve some kind of a medal actually for working with me, but this is a project from hell because every country has its own soil classification system and they don't match up. And so he had to go into Brazil and get the Portuguese soil orders and figure out how that related to everybody. It was a really hard project. But he pulled together this basically global nutrient atlas. It's really cool. And that's what I showed you, that phosphorus map is one of the products of that. And one of the things you can do with that, so this is the phosphorus map, the results from, so a lot of work went into this, and also not only matching up different soil taxonomies, but then what level of phosphorus is enough for plant growth, and people have different ways to test that, and it was a really complicated project. So from that analysis, you can do a kind of a GIS type analysis saying where are the plants, where are the soils, and so on. So this is a Venn diagram. This circle represents the vegetation of the Earth, and then this circle is the low phosphorus areas of the Earth, and from which you can see over half of the plants on Earth are growing in low phosphorus soils, half of the vegetated area of the Earth. This is the area expected to receive a large temperature increase this century. And so you can see that over half of the land that's going to be affected by high temperatures, according to this climate scenario, these climate scenarios, are actually on low phosphorus. What that means is, to understand how plants are going to respond to temperature change, we have to understand how temperature change interacts with phosphorus, because that's what a lot of plants have to deal with, the mineral stress of phosphorus deficiency, as well as, in this case, a temperature change. So we take these aggregate scenarios. This is the IPCC Global USA mid-range scenario. You guys have a wonderful seminar series, and I think you probably heard about some of these scenarios, right, during the series. So I won't go into detail. The warmer colors is where it's going to get warmer, obviously. And so we can interface that with our phosphorus map and say these are the areas that are going to get very warm that are also very low phosphorus. The Amazon jumps out. Utah's in there someplace, Western Europe, you know. So these are the low phosphorus vegetated areas that are expected to get really warm. And then, so we thought, okay, temperature affects phenology, has a huge effect on phenology, the timing of when plants germinate, when they flower, and when they set seed for annual plants. And for perennial plants also, when they flower, how they grow and reproduce is very much affected by phenology in many cases. And so Eric and I were thinking about this, saying, well, let's just say that, you know, through a growing season, everything is fine, and eventually it gets dry or cold or something hot, something happens, and your risk of mortality increases. Here in Utah or in Pennsylvania, come September sometime, you're going to die because it's going to get really cold if you're a corn plant or an Arabidopsis plant. So if you grow and then flower and set seed, you should finish that up before things get really rough. And that's what we see plants trying to do. If you shift the mortality curve, then you're able to get a plant that grows longer, develop more biomass, and set seed and have more yield. Having a longer growing season is going to give you more yield, all else being equal. And so that extra, you know, so that duration of the acquired resources increases, and so you have more output. Does everybody follow me on that, or do I have to explain that a little more? I guess some of you aren't scientists, is that right? Let me know if you want me to explain something. I'm going too fast. So we predicted that a delayed phenology was going to help us in low phosphorus environments for two reasons. One, it was going to give the roots longer to pick up phosphorus, and the other is once you had phosphorus, it was going to give the leaves longer to use that phosphorus. So here's an example from computer simulations. Here's a root system, and the zones around the roots represents the zones of diffusion where phosphorus has diffused to the root. This is a sophisticated computer simulation. These aren't just pictures I drew. And so it shows you that over time those diffusion zones expand because diffusion is slow, but it proceeds over time. So basically, if we have more time of the roots being in the soil longer, we can go from this kind of a scenario to this kind of scenario. There's time for more phosphorus to diffuse to the root. And so this is Eric's study. It was Arabidopsis, little Arabidopsis plants growing in natural soil that's low phosphorus. And what he found was this is a log scale of root length duration, how long the roots were in the soil. And this is a log scale of how much phosphorus they took up. The blue is our high phosphorus plants. The red is our low phosphorus plants. We saw a very close correlation between how long those roots were out there in the soil and how much phosphorus they took up, obviously. You can see it here. In terms of reproduction, high phosphorus plants, there was really no pattern in terms of yield, seed yield versus how long they spent growing. But under low phosphorus, there was a very sharp pattern. The longer those low phosphorus plants could grow, the more seed yield we got. In fact, because we were working with the Arabidopsis, we could use genetic mutants, and we had mutants that had delayed flowering, so they had a longer growth cycle. So here we have normal plants, and we're looking at vegetative biomass, reproductive biomass in red. So under high phosphorus, they flowered normally at 31 days. That was delayed 5 days by low phosphorus. They matured 73 days normally. Low phosphorus delayed that. So low phosphorus makes plants slow down. So I think plants know what they're doing. Now, these mutants had even longer delays because of the genetic mutation. So they had actually quite a bit longer to grow under those low phosphorus conditions. And under low phosphorus conditions, they had no reduction in yield. The wild-type plants actually reduce yield by low phosphorus. These delayed plants, artificially delayed plants, had no yield reduction because of low phosphorus. Here's another line of evidence. Eric made a very complex computer model simulating carbon and phosphorus dynamics, including senescence and mortality. I'm not going to go into the details here, but I'm going to show you the key result. This is initiation of reproduction in days. This is the length of how long it takes the vegetative growth to stop once you start reproducing. And he found that under high P, your best strategy was to grow for 38 days, whereas under low phosphorus, your best strategy was to grow for 50 days. So whether it's with mutations or natural phenotypic variation or modeling, the message is if you have longer, if you have more time, you'll do better on a low phosphorus soil. So why do I bring this up? Is that altered growing seasons, which we have already seen as a result of climate change and expected to increase in the future, will affect soil resource acquisition and utilization. We can expect earlier springs in the temperate zone, longer falls, but it may be a dry fall, so that mortality function is not certain. Later maturing taxa can acquire more of these diffusion-limited nutrients like phosphorus and use them more efficiently. An extra day of growing with that phosphorus is an extra day of photosynthesis you can pull out. The result could be reduced P stress in high latitudes and increased root competition in the tropics. Remember that the Amazon was one of the places where we're concerned about both temperature change and low phosphorus soils. We really don't know what's going to happen in a place like that. But the main point is effects of altered phenology on mobile resources such as nitrate and water could be quite important, but they're poorly understood, especially when your plant altered phenology does not synchronize well with the altered rainfall distribution or something like that. You can imagine here in Utah the snow is melting earlier, are your plants going to be able to take advantage of that, and these kinds of things. This kind of synchronization is very important. Climate effects on plant phenology will have large and unknown effects on soil resource capture and utilization. Third example. This is Professor St. Clair's doctoral work. He didn't want me to show this because he thought it was old news, but I think it's very cool. He wrote a lot of really good papers, and some of those are still coming out, so there's new aspects of this that even Professor St. Clair has not seen. And this is climate interactions with manganese toxicity in the eastern forest. So this is the eastern forest of North America. We were doing this work right around here, and so this is the middle of the eastern forest. This is what it looks like in the fall, and that red color, a lot of it's driven by maples, and sugar maple in particular has a beautiful color, and of course you make maple syrup, and it used to be a dominant species in the eastern forest, and it's losing its dominance, being replaced by other plants like red maple. And this is what you'll see in these sugar maple stands, this kind of crown dieback. So this tree has actually died. You can see this one is very sick. This one is dying back at the top. This is dead. So a healthy stand should be solid closed canopy. We see this blue sky. We know that there's this sugar maple decline. This is the range of sugar maple. This is also happens to coincide fairly well with acid deposition, which may be one of the driving factors. As you decrease soil pH because of acid rain and logging and other things, the manganese in the leaves goes up dramatically. So this is kind of an acid soil issue. Study by Steve Horsley and colleagues, year 2000, did a kind of broad survey across the Allegheny Plateau looking at what was associated with sugar maple decline, and found that it was associated with foliar levels of magnesium, manganese, and defoliation. Okay, so here's was our idea that Sam and I were working with based upon some other stuff we've done with working in the tropics, that this is the Z-scheme, the photosynthetic electron transport, and there's various metal cofactors throughout this. You see here's actually manganese, which is involved in producing the oxygen that you're breathing, and magnesium and chlorophyll, and iron and copper are involved in electron transport chain, and so on. And if electrons flow through this normally, great. But if there's something blocking one of these steps, those high energy electrons will not know what to do, and they'll end up probably jumping onto oxygen, which is being produced right there, and making these very reactive, very damaging free radicals, these oxygen radical species, which can be detoxified once again with enzymes that have metal cofactors. So metals are really important in terms of making sure those electrons are going in the right place and they're not causing damage. Now, so the idea was manganese, which can form reactive oxygen species itself, and then by interfering with these metals through competition, could disrupt the normal pathway and disrupt the detoxification of these free radicals. That was an idea, that maybe manganese is is a key player in this forest decline that we're seeing with sugar maples. This is Sam's data. So he looked at a range of tree species, each one shown in this abbreviation. Sugar maple is circled in red. And here he's looking at increasing levels of foliar manganese. And then here we're looking at chlorophyll and electron transport and stomatal conductance and CO2 assimilation. And in every case, sugar maple seems to be accumulating a lot of manganese, and it's below the line, which means it's more sensitive than the other species to manganese. Okay, so that was interesting. So basically our idea was that manganese is getting into the sugar maple, and in response to high light, it's causing the formation of these damaging species, which then causes toxicity. That was a simplistic assumption going in. So we go to these stressed stands. Sam identified different regions in Pennsylvania that had these forest decline symptoms. Here he is with this Penn State gene gun, right? Because we were originally going to try to get genetic transcripts out of this. Didn't actually work out. This is actually a $60 shotgun from Walmart, I think is what it was. And so this is what it looks like. Sam actually took his professor out into the woods at one point to let me see what it was like, and I couldn't hit a tree. I guess you got to sneak up on him just right or something. I didn't. But he would aim for a particular branch up there and catch it before it hit the ground. Now, I understand that Sam has graduate students who can catch 2 branches at once with 2 hands. So I think things are getting better with this methodology. So before that branch knew it was disconnected from the mama tree, it was being put into the photosynthesis machine, and he was getting data. And here he is. Okay. So from this kind of data, you see, look, the more manganese you have in those leaves, there's a very tight correlation with reducing photosynthesis. This is one of the things that convinced Sam it was a worthwhile thesis topic. There's really something going on here with manganese. So he also did some controlled environment studies. You go to one of these stressful sites like this upper slope position here in an acid soil, very carefully collected soil so that he separated the horizons and reassembled the horizons in a pot after removing chunks of debris and stuff to capture the chemistry of manganese, how it's reduced and oxidized in the soil. And then he could grow them with shading or normal light in the greenhouse to see how light interacts with this acid soil situation. And here you can see sugar maple leaves at normal manganese and high manganese, and he's put a piece of filter paper on the leaf. So you can see where the shade of the filter paper was, the leaves are still green, and where the light has been hitting the leaf, you can see there's damage, there's bleaching, there's loss of chlorophyll. And you can see that here in the data here. He's got highlight and shade. The blue is normal manganese, the red is high manganese. And he's looking at photosynthesis and electron flow and chlorophyll. And in each case, the worst damage is shading. The manganese doesn't cause too much damage in a shaded plant. In a highlight plant, causes lots of damage. This is comparing red maple with sugar maple. Red maple is a species that's replacing sugar maple. And if we're looking at photosynthesis on these more stressful soils and the upper slope versus the better soils, the lower slope, red maple really doesn't care. It does well in any slope position. Sugar maple, very sensitive to slope position. It's very sensitive to those bad soils you find up at the top of the ridge. And this may be one reason why red maple is replacing sugar maple, is it can tolerate these stressful soil conditions. Sam did a liming study where he raises the pH. Red maple didn't care. Sugar maple, very sensitive to changing the pH. And he found an interesting relationship between mycorrhizal colonization in the sugar maple and then photosynthesis. You can see here we're measuring infection percentage of the mycorrhizas, and we're looking at red maple, sugar maple. So as we increase pH, sugar maple has really good mycorrhization. At low pH, it has very poor mycorrhization. In fact, he showed that acid soil is as effective as fungicide in reducing AM colonization. Here we're looking at, once again, mycorrhizal colonization, photosynthesis, electron transport. With fungicide is red. So in this acid soil, it's like we've given a happy plant fungicide. We'll get the same colonization we have in acid soil. So there was this kind of series of papers. I won't go into a lot more detail on this, but a story developing about manganese affecting photosynthesis and the carbon limitation perhaps affecting mycorrhizas. The mycorrhizas were not happy, not providing enough phosphorus to the leaves. So there was this kind of a vicious cycle of low carbon, low phosphorus affecting these plants that seemed to be implicated with manganese getting into the leaves. So there's several things going on in these acid soils, of course, but manganese seems to be an important player. And it seems to be interacting with light to create oxidative stress, which is interesting from a climate change perspective, because there's a lot of things that are part of climate change that affect this formation of reactive oxygen species. Ozone, UV radiation, light intensity itself, temperature, acid deposition affects the chemistry of the soil in a way that makes manganese more available. Rainfall makes manganese more available. So it's a complex kind of set of factors that's not necessarily changing the amount of manganese in the soil, but it's suddenly making it more available, and when it gets into the leaves, more toxic. And I think this is a great, one reason I like this is it's a beautiful story, a beautiful piece of work that Sam did, I think. But it's also a very good example of this kind of wild card I meant. It's not a linear type of response where you can say, oh, higher CO2 means higher this. No. Here's a changing environment that's making something that formerly wasn't toxic, now all of a sudden very toxic and having multiple interactions. Ozone, for example, if we're correct that ozone is part of this dynamic, we should be able to test that. So here's sugar maple seedlings growing in little chambers that have either UV passing or UV filtering mylar. And what we see is that here's with UV, and here's farm soil, good soil. Doesn't matter if they have UV or not. In a forest soil, that UV is causing this chlorosis. I hope you can see that in this picture. So here again, we're giving an interaction between soil conditions, mineral stress, and a factor that's part of atmospheric change. And I want to talk about this little guy. This is gypsy moth, well known to anybody who's visited the eastern forest, especially in some years. You get you can hear the frass hitting the ground, and there's some massive defoliation. Remember the Horsley study indicated that part of the sugar maple decline syndrome was defoliation, okay? Defoliation as well as manganese and magnesium. Now this is work that was led by Jennifer Dean as part of her PhD with the help of Consuelo de Moraes and Mark Mesher. So they were looking specifically at the interaction of manganese and this defoliation. Why would manganese stress be related to defoliation, or were they separate processes? So, of course, you know, the idea is herbivory is affected by many things the plant does to try to protect itself. And our notion was that somehow acid soil stress was affecting this defense pathway in a way that was making the plant more sensitive to herbivory. That was the idea. And so we were looking at defenses the plants were making and tannins, and also signaling molecules that are involved in telling the plants it's time to protect yourself from herbivory. So Jennifer had a series of questions. Do herbivores prefer to feed on sugar maple when it's stressed? Do they grow better on stressed sugar maples? Are there differences in the phenolic compounds, defense compounds in the sugar maples? And can the sugar maples induce these signaling molecules to protect themselves when they're attacked? So these are the things that she looked at. So here she's looking at gypsy moth. And so she gives them either a bit of a leaf from a stressed plant or from a healthy plant, and puts the caterpillars in the petri dish and sees what they prefer to eat. And they actually preferred to eat leaves from the stressed plants. Oddly enough, you'd think a healthy leaf would somehow be more nutritious, but in this case, they actually preferred to eat the stressed leaves. Do they grow better when they eat those leaves? So this is the study. We put little mesocosms out, and then she bagged them at some point and put the caterpillars on there. There's some of those little devils right there. And then at the end of the day, you can say, well, healthy soil, here's what they look like. Acidic soil, they obviously preferred, right? And she can look actually at the performance of the larvae. And she found that if they were undamaged, it was about the same. But if we had a previous damaged leaf, the healthy soil plants, the caterpillars didn't grow very well. Whereas on the acidic soil grown plants, the caterpillars grew fine. So it's like these plants could protect themselves, had some warning because they were previously damaged and could protect themselves, and these plants couldn't do it. So looking at the tannin story, we found generally more of these kind of passive defense molecules in the acidic soil-grown plants, which is what I would have expected, which is kind of strange the caterpillars will have preferred that. But in looking at these induced defenses, so she went out into the forest and did experiments with plants in the forest and froze tissue, brought it back to the lab for analysis. Here's the jasmonic acid, which is a key signaling molecule in terms of plants protecting themselves from herbivory. And so you can see the plants that are grown in the acidic soil cannot make as much of this signaling molecule. And instead, they make more salicylic acid. And this is like an alternative signaling pathway which may protect you from abiotic stress and from pathogens, but doesn't seem to help with herbivory. So this seemed to support the hypothesis that the manganese was actually predisposing the plants to caterpillar attack. The caterpillars preferred the stress leaves. They grew better on the stress leaves. The stress interfered with the ability of the plants to induce defenses against the caterpillars. So what are the points I want to make from this? Manganese toxicity is something most people don't think about. It's kind of a mineral that is of interest to plant nutritionists, but not a lot of people think about it too much. Is an important factor in the health of these trees in the eastern forest. It's associated with photooxidative stress, meaning toxicity exacerbated by high light and other components of climate change. We saw reduced mycorrhizal effects, which may intensify the nutrient stress, had diminished herbivore defenses in response to the stress, and more attractive to gypsy moth larvae. We predict that because of ongoing soil acidification from acid rain and logging and increased oxidative stress, warmer, brighter years, and of course that's autocatalytic. As you begin to lose trees, now there's more light coming in, and the leaves that are left have more light to deal with. We're going to see increasing manganese toxicity in years to come, is our prediction, partly as a result of climate change. And this, I think, is a good example of nonlinear interactions between nutrient stress, which I believe, if we look, we will find to be more common. That instead of saying high CO2 is going to help plants, it's like, well, maybe not. Or, you know, plants will have more photosynthesis and be able to deal with manganese stress. No, actually, you know, high light or high temperatures may make manganese stress much worse, and then may make insects come in and make problems even worse for the sugar maple. And we also see the genetic variation in manganese response, in this case between red maple and sugar maple, is driving a species shift in this forest, which could be quite significant if we go from something like a broadleaf to a conifer. And of course, this is important because, once again, going back to the Venn diagram, this is the total vegetated area of the earth, and this is acid soil, and this is expected high temperature change, just as one example. And you can see a significant portion of the plants that are going to see a high temperature change are actually growing on acid soil. The fourth thing I want to bring up, final thing I want to bring up, is we don't have a good working model about how plants deal with multiple stresses. Most of the literature is about how plants deal with one thing. There's not a lot of studies where they look at two things. And there's two paradigms out there for how plants deal with multiple resource limitations. One is very old. It's called the law of the minimum. It's usually referred as Liebig's law. We now know from a little bit of historical research that Liebig was actually stealing the results of a younger scientist by the name of Sprengel. So it should be called Sprengel's law, but it's the law of the minimum, 1828. Basically, this is in the early days of fertilizer chemistry and soil fertility, and they were saying plants will respond to the most limiting resource. You can give a plant tons of nitrogen. If it doesn't have potassium, it won't grow. You need to give it potassium for it to grow. So the law of the minimum is you need the thing that's limiting to grow. That's one prediction. That would predict elevated CO2 will help plants. Because, I'm sorry, it will not help plants because if you don't have nitrogen, that extra carbon won't help you. Now, in contrast, the microeconomic paradigm would say it would help plants. This is, I think, embodied by Mooney and his students, and this is the idea as kind of an economic analogy of plants, that if they have a lot of one resource, they can somehow trade that into more of another resource. If you want to make a cheese sandwich and I give you a truckload of cheese and no bread, you're going to trade or sell some of that cheese and get some bread and make your sandwich. You can turn the extra cheese into a cheese sandwich. So he would say, they would predict, and they have predicted in writing, that elevated CO2 is going to help plants get water and nutrients and everything else, because now you have more sugar, you can grow more leaves and roots, and plants are going to benefit. One resource can be traded into another. Now, these two paradigms have different predictions about how plants respond to multiple stresses. One says the limiting resource is going to drive it, and one says, no, you can interchange resources. So this is work that was done by Gerardo Rubio about 10 years ago to test this. He looked at all pairs of nitrogen, phosphorus, potassium, magnesium, and all different combinations to see if plants behaved according to Liebig or Mooney. So you have Liebig on one end of the spectrum and Mooney on the other. And somewhere in between is how plants actually live. So if we look at nitrogen stress, nitrogen believes in Hal Mooney. Nitrogen acts like an economic resource. If you give a plant more nitrogen, it can do all kinds of fun stuff with that and go out and get other things, just like Mooney would predict. However, potassium doesn't believe in that. Potassium believes in the law of the minimum. If a plant does not have potassium, nothing else you can give it is really going to help. It needs potassium, which is kind of interesting because Hal Mooney, most of his literature, when he talks about nitrogen or nutrients, it deals with nitrogen. And these guys were very interested in potassium. So it's kind of interesting how their things that they studied affected their theories. However, magnesium doesn't obey either. It depends upon what it's in combination with. Sometimes it behaves like a Liebig nutrient. Sometimes it behaves like a Mooney nutrient. And phosphorus really didn't obey anything. So the point is, as scientists, as human beings, we're always looking for patterns in nature. We're trying to simplify things. In this case, you can't do it. There is no simple model that applies to everything. If you're interested in how plants deal with low magnesium, you better study low magnesium. You can't assume that high CO2 is going to help a plant with magnesium. That may not be true. And this is just a conceptual failing we have in our discipline. So the points I tried to make today, most plants suffer at least one mineral stress, usually multiple. Little is known about how these stresses, mineral stresses, interact with climate change, but they're likely to be important, these interactions. I gave you the example of soil taxa not responding to elevated CO2. I gave you the example of plants that can grow longer, solving their phosphorus problems. And I gave you the example of manganese, where things went in an entirely unexpected direction because something that was always there now suddenly becomes more toxic because of atmospheric variables. And then I told you we really don't know how to think about these things very well. So you may not find this to be a very satisfying seminar, but this is where we're at. My conclusion is that this is a wild card in plant responses to climate change that we have to think about more. So there's a lot of people we have to thank here. This is Steve Horsley, USDA Forest Service, Bob Snyder, technician, some of the entomology crew. And of course, the main players for the soil taxa story, Eric and phenology. Here's Professor St. Clair in his salad days, manganese, and the Liebig Mooney is Gerard Rubio and Jinming Zhu. Thank you very much.